Hi everyone. In the immunology subject, now we are going to learn about hypersensitivity. Generally, we call it as allergy too. An immune response generally evokes a battery of effector molecules that act to remove antigen by various mechanisms. That's what happens normally. If these effector molecules may include subclinical localized inflammatory response, these are all going to eliminate without causing any damage to the host tissue. But certain circumstances results this inflammatory response can have deleterious effects and resulting in our tissue damage or even death. This inappropriate immune response is going to be termed as hypersensitivity. So this hypersensitivity can also be, in simple terms, we can also say it as too much reaction of the immune system. If the immune system is going to react too much, then we can simply define it as a hypersensitivity. So let's begin with the definition. So hypersensitivity can be defined as an unexpected, exaggregated, adverse reaction to an antigen or hapten. That means the foreign particles that is entering into the host. If our body is going to respond in an exaggregated adverse reaction, then we call that reaction as hypersensitivity. This can also be defined as it is an exaggregated immune response that results in tissue damage and is manifested in the individual on or subsequent contact with an antigen. There is no need to respond immediately when the antigen comes into the host. So it may occur frequently exposing or sometimes immediately after the entry of the antigen. Such type of an exaggregated immune response is going to be called as hypersensitivity. And coming to the classification or types of hypersensitivity, basically hypersensitivity is going to be classified into two types based on time of reaction and based on the different mechanism of pathogens. So the first coming to the based on the time taken for the reaction, the hypersensitivity is going to be classified into two types. One is the immediate type, another one is the delayed type. The name itself is indicating if you are going to have the reaction immediately after the entry of the pathogen, then we call it as immediate type. And these are all the different types of immediate type of hypersensitivity. Then coming to the delayed type, so where the reaction takes place of more than 48 hours, then we call it as a delayed time. And this contact sensitivity, allergy to various microorganisms, tuberculin allergy, rejection of foreign tissue graft. So these all will take nearly more than 48 hours to have the hypersensitivity. So such type of the reactions were classified under delayed type of hypersensitivity. So these are the two types of the hypersensitivity based on the time taken for the reaction. Then coming to the based on the different mechanisms of pathogens, we had basically four types. Recently, a fifth type was also included. But according to the Coombs and Gell, they proposed the four types of hypersensitivity. That is type 1, type 2, type 3, type 4. The type 1 is also called as anaphylaxis. Type 2 is also called as cytotoxic. Type 3 is also called as immune complex hypersensitivity. Type 4 is going to be called as cell-mediated hypersensitivity. And the fifth one is going to be called as stimulatory type reaction. Okay, so let's see all the types of hypersensitivity in detail now. So coming to the immediate type hypersensitivity and the delayed type of hypersensitivity, here just I have mentioned some differences. So let's have a quick of these points and then we will move to the types of hypersensitivity. So as I told you, the immediate hypersensitivity is going to manifest in a short duration of time within the minutes. But coming to the delayed type, it will slowly manifest from 24 hours to 72 hours. Then here the immune reaction appears and disappears rapidly. Okay, that But in the delayed type, the immune reaction appears slowly and it is going to last longer. Then the inflammatory response occurs in a few minutes. That means you are going to see the swelling nature, all the things. But here the inflammatory response manifests only after 24 to 48 hours. And the immediate type is going to produce urticaria, wheal and granulocyte accumulation, whereas the delayed type is going to produce the erythema, induration and lymphocyte infiltration. Then the immediate type hypersensitivity is going to involve in antigen and antibody interaction, but the delayed type is going to have a reaction between the antigen and T cells. So we can consider this as a cell mediated. Then coming to the immediate, it can be passively transferred from one host to the another by the transfer of serum, but the delayed type cannot be transferred from the sensitized host to another by the transfer of T cells. And the desensitization can be easily done by a small dose of allergen, but the desensitization cannot be easily done by drugs. And this is going to be, that means immediate type of hypersensitivity can be suppressed or inhibited by antihistamine drugs except the for the reaction called as Arthur's reaction. Then here this delayed type hypersensitivity can be suppressed by the drugs called as corticosteroids. So this is a difference between the immediate type hypersensitivity and delayed type hypersensitivity. If the question is asked regarding the immediate type hypersensitivity, these all points separately you can write for the immediate hypersensitivity. In the same manner, if the short question was given as a delayed type hypersensitivity, you can write all these points as a separate for the delayed type hypersensitivity. Now let's begin with the Coombs and Gell classification, where they have classified the hypersensitivity into majorly four types. Type 1, which is also called as anaphylaxis, then type 2, which is also called as anti-mediated hypersensitivity, type 3 is called as immune complex mediated, and the type 4 is called as delayed type hypersensitivity or cell mediated hypersensitivity. Let's begin with the first one, type 1 hypersensitivity, which is also called as anaphylaxis or allergic hypersensitivity. Then what is an anaphylaxis? How can you define this? It is defined as an exaggregated reaction of an organism to a foreign to which it has previously become sensitized, resulting from the release of some chemical substances like histamine, serotonin, and other vasoactive substances, is going to be called as anaphylaxis. And the nature of the antigen, the type 1 hypersensitivity, is soluble. That means the antigen will be soluble and it is going to be exogenous, but is coming from outside, and it will be free in circulation of blood. Then coming to the antibody involved in this hypersensitivity reaction is IgE, and which is going to fixed on mast cells and basophils. And then moving to the mechanism, allergen-specific IgE antibodies bind to mast cells via their Fc receptor. When the specific allergen binds to the IgE, cross-linking of IgE induces degranulation of mast cells. Example, local and systemic anaphylaxis, seasonal hay fever, food allergies, and drug allergies. So let's see this mechanism in detail now. So here is a picture of what it is happening. So for example, this anaphylaxis is going to be mainly characterized by an allergic reaction occurring immediately following an individual's second contact with the responsible allergen. Upon the initial exposure, that means when an allergen enters into the host, and what is happening, these B cells, that is these antigen presenting cells along with this antigen, will be stimulating the B cells, and B cells are stimulated, okay, and they are getting into the plasma cells and produce specific IgE with the help of T cells. Now this is what happening in the first exposure. This IgE is sometimes being called as reagent, that we have discussed already in the types of antigen antibody reaction. Now the individual has a hereditary that is disposition for its production. Once synthesized IgE binds to this B cell and to the receptors of basophils or that is mast cells, and sensitizes these cells, making the individual allergic to the allergen. So this is what happening in the first exposure. That means when an allergen enters into the host body, that allergen is going to be taken up by the antigen presenting cell, and now the B cell is going to be stimulated with the help of the T cells and releases the plasma cell, and in the circulation we are going to have the IgE immunoglobulins. And now these IgE immunoglobulins will go and bind to the mast cells and the basophils. So these are all things happening in the first exposure. Now, when the second exposure is going to occur, that means with the same antigen or allergen occurs, the allergen directly attaches to the surface bound IgE on the sensitized mast cells, causing degranulation, and these degranulation releases some sort of physiological mediators such as histamine, leukotriene, then slow reacting substances of anaphylaxis called as leukotrienes, okay, then heparin and some sort of prostaglandins and platelet activation factors, then eosinophil chemotactic factors of anaphylaxis and proteolytic enzymes. So these are some of the physiological mediators that are being released because of this degranulation. Okay. Now these mediators, what these mediators will do is they trigger the smooth muscle contractions, and then vasodilation occurs, increased vascular permeability and mucus secretions. The inclusive term of these all responses is going to be called as anaphylaxis. So remember the inclusive term of all these reactions, that is the triggering of smooth muscle contractions, vasodilation, all these things is going to be called as anaphylaxis. So this is all about the type 1 hypersensitivity, and here you have to remember the definition, nature of antigen, antibody involved, mechanism of activation and the examples of type 1 hypersensitivity. Then moving to the type 2 hypersensitivity. So this type 2 hypersensitivity is also called as cytotoxic hypersensitivity, which is also going to be called as antibody dependent cell mediated cytotoxicity, simply called as ADCC. And here the nature of antigen is cell bound antigen, then it's an exogenous or endogenous, then fixed on cells. And which type of antibody is involved here is IgM or sometimes IgG, but majorly IgM is going to be involved in this type of hypersensitivity. And obviously the IgM or IgG is free in circulation. Then coming to the mechanism of activation, how this hypersensitivity is getting activated, let's see. So that this type of hypersensitivity is initiated by cell surface bound antigens. So here you can see the cell surface bound antigens, and is mediated by mainly IgG antibodies, sometimes IgM also. The antibodies obviously bind to the antigens which are present on the cell surface, isn't it? Now, this immune complex, that is antigen antibody complex, activates the complement system, okay, or cytotoxic T cells, leading to the ADCC, that is antibody dependent cell mediated cytotoxicity, and leading to the destruction of what it is going to destruction of the target cells. The effector molecules in the complement reaction are going to be of our C3b or C4a or C5b. So this type of hypersensitivity is going to be called as cytotoxic, that is antibody mediated cytotoxic hypersensitivity, or simply called as cytotoxic hypersensitivity. So here the examples included in this one is going to be the blood transfusion reactions, erythroblastophetalysis, autoimmune hemolytic anemia. Okay, so this is all the mechanism that we had gone through this in this second type of hypersensitivity. Then moving to the third type of hypersensitivity, which is also called as immune complex hypersensitivity. Then coming to the nature of antigen, it's a soluble antigen, okay, exogenous or endogenous and free in circulation. And the antibody involved is both IgM or IgG, but here it is going to be of IgM, and previous one it is IgG. Then moving to the mechanism of activation, antigen antibody complex are deposited in tissues, And complement activation provides inflammatory mediators and recruits neutrophils. And enzymes released from neutrophils damages the tissue. So let's see the mechanism. So as I told you, this type of hypersensitivity is induced by soluble antigen and is mediated by IgM antibodies that interact with the antigens to form an that is antigen antibody complex. Now this antigen antibody complexes, that is immune complex, become deposited in various tissues and induce complement activation. So they are going to be deposited in the tissues and they are going to activate or induce the complement activation. This induction or induces an inflammatory responses mediated by massive filtration of these neutrophils in the circulatory system. So that's how, as these neutrophils are coming and doing all these things, then obviously the cells are getting to be burst out, and that's how it is going to release all the inflammatory mediators like histamine, all these things, and obviously are going to have vasodilation, everything. And the examples of this kind of hypersensitivity are going to be serum sickness, rheumatoid arthritis, then systemic lupus erythematosus, post-streptococcal glomerulonephritis. So these are the examples of the third type of the hypersensitivity, that is immune complex mediated hypersensitivity. Then moving to the next fourth type of hypersensitivity, that is a cell mediated hypersensitivity or delay type hypersensitivity. So here again the nature of antigen is soluble, exogenous or endogenous, and the antigens are free in circulation. But coming to the antibody involved, here no antibodies are going to be involved. That's why we call it as a cell mediated hypersensitivity. Okay, and here you can see mainly the subsets of the CD4, that is cluster differentiation of some helper T cells or cluster differentiation of 8 cytotoxic T cells are involved in the type 4 hypersensitivity. And the mechanism of the activation of these cells is going to be of T cells secrete cytokinins which activate the macrophages, and that's how cytotoxic T cells will lyse the cells. Okay, so let's see the activation mechanism here. So as I told you, the type 4 hypersensitivity is initiated by soluble antigens, okay, and then the TH cells, that is T helper cells. So here is our T helper cell. The activated T helper cells release some sort of a cytokinins that activate the macrophages or TC cells, that is T cytotoxic cells, and which mediate the direct cellular damage. And the activated T cells secrete various cytokinins which act as effector molecules in this type of hypersensitivity. So here the T cells are going to secrete some sort of cytokinins, and these cytokinins will what we'll call it as induce the activation of macrophages, and these macrophages will directly damage and secrete some sort of chemical substances which are going to damage our cells, and that's how causing the cell mediated hypersensitivity. And the examples coming under this type 4 hypersensitivity is going to be of mainly contact dermatitis, graft rejection, tubercular lesions, and multiple sclerosis. Okay, since it is going to take much time to manifest the symptoms, we can consider this also as delayed type hypersensitivity. Okay, so this is all about the 4 types of the hypersensitivity that is going to be given according to the Coombs and Gell classification. And recently, a 5th type of hypersensitivity was also introduced into the types of hypersensitivity, that is stimulatory type reaction. So the example of this kind of stimulatory type of reaction is Graves' disease, in which thyroid hormones are produced in excess amount. So actually, initially it was classified under the type 2, but later on some sort of the differences was there between the type 2 and this type 5, that is stimulatory. So obviously it was classified under another type called as type 5 hypersensitivity. And the best studied of stimulatory type of reaction is a Graves' disease. Thyroid stimulating antibody is a specific thyroid or autoantibody to thyroid membrane antigen. So look at this picture. In general, the pituitary gland is going to release the thyroid stimulating hormone, and this stimulates the hormone synthesis, and you are going to have the thyroid hormones release. And if they are going to be excess, these are going to have the negative feedback and tells the pituitary gland to stop the thyroid stimulating hormone. In general, this is what happening. But in the stimulatory reaction, what is happening is, that is especially in the Graves' disease. So an antibody against this thyroid stimulating hormone is going to be generated or stimulated, and that will cross the placenta, which lead to the hyperthyroidism. So how this antibody crosses the placenta and binds to this receptor, that is TSH receptor. Actually, the thyroid stimulating hormone from the pituitary have to bind to this receptor. But instead of that stimulating hormone, this antibodies, autoantibody is going to bind to this TSH receptor. Now what happens? This stimulates the hormone synthesis, and you are going to have a unregulated reproduction of or overproduction of thyroid hormones, which leads to the hyperthyroidism. So that's how this is going to be leading it to a disease called as Graves' disease. So it is pre-assumed, as I told you, it's the second type. Combines with a site on the TSH, resulting in excessive secretion of thyroid hormone. The thyroid stimulating hormone can cross the placenta, which lead to hyperthyroidism in neonat. Neonatal hyperthyroidism subsides within a few weeks after birth with degradation of maternal IgG. So this is all about the 5 types of hypersensitivity that we had gone through. Let's have a quick review of all the types of mechanisms of hypersensitivity in the form of a flowchart. So here you can see the nature of antigen, the type 1, type 2, type 3 and type 4. Soluble, it is cell surface bound, here again soluble and here soluble. And the type of the antibody involved in the type 1 is IgE, type 2 is IgG, type 3 is IgM and type 4 no antibody, it is a cell mediated. Then type of immune response, obviously antibody involved humoral, humoral and humoral, whereas this is cellular. Then the effector molecules that are being released because of these hypersensitivity are histamine and other biological active molecules. Whereas here in the type 2, it's membrane attack complexes and the complements like C3A, C4A and C5A. Then type 3, here the effector molecules are neutrophils, and in the type 4, the various cytokines secreted by our activated T cells. And coming to the mechanism, which is very important, B cells are going to secrete the immunoglobulin once they are getting activated by the allergen, that is IgE. And now this IgE binds to the Fc receptor on mast cells and blood basophils. Now coming to the second exposure of the same antigen or allergen, it cross-links with the IgE, activates the mast cells and blood basophils and secretes the vasoactive amines, and these are going to be involved in contraction of smooth muscles and increasing the vascular permeability, vasodilation, everything occurs. And the examples of the type 1 that we have discussed is systemic anaphylaxis, localized anaphylaxis, and hay fever, asthma, food allergies, everything comes under the type 1. In the same manner, we have discussed about the type 2 hypersensitivity, where here the IgG antibodies are involved. So here the B cells activated by antigen secretes the IgG, and this IgG binds to the cell surface bound antigen, activates the T cells and then activates the complement system. That's the reason why we call this as anti-mediated cytotoxic hypersensitivity. And obviously they secrete various cytokines leading to the ultimately cell death. And activated complement system leads to the cell death. T cells also leads, and the complement system also leads to the cell death. And the examples of this kind of type 2 hypersensitivity we have discussed is blood transfusion, erythrobastrophetalysis, then autoimmune hemolytic anemia. Then the third type of hypersensitivity, that is immune complex mediated hypersensitivity. So here comes the B cells, upon activation with the antigen secretes an antibody called as IgM. Now this IgM interacts with antigen to form immune complex, and now the immune complex forms, the complement system getting activated and causing to the inflammatory response, massive infiltration by the neutrophils. So here the effector cells are the neutrophils. Then what are the examples under this? We have studied these: serum sickness, rheumatoid arthritis, systemic lupus erythematosus. And coming to the fourth type of hypersensitivity, that is going to be cell mediated hypersensitivity, where no antibodies are involved. So here what are the cells that are involved? Are the T helper cells. Okay, now these T helper cells, upon activated with the antigens, secrete some sort of chemical substances called as cytokinins, and these cytokinins gets activating the macrophages and C, that is cytotoxic cells, that is T cytotoxic cells, and these will initiate the phagocytosis by macrophages and various cytokines secreted by the T cells. And that's how the cellular damage is going to occur. So this is the overall view of the four types of the hypersensitivity that we have discussed. Okay, so this is what we have discussed about the four types of hypersensitivity. So you can go through overall the tabular form and this the four types of the hypersensitivity diagrammatical representation at a glance, and if you write as a short also, easily you can gain more marks. Okay, thank you.